Today's educational web seminar is presented by LabRoots and brought to you by Biosearch Technologies. Hello everyone, and welcome to the Coronavirus webinar series produced by LabRoots. Today's webinar title is Rapid Response to the Coronavirus Outbreak, How Collaboration Meets the Challenge. I am Xavier Gutierrez of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. And as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Geert Koene. Dr. Koene holds a PhD in immuno-oncology from Utrecht University in the Netherlands and has been active in the molecular diagnostics industry for over 15 years in various global roles. He joined LGC Biosearch Technologies 1.5 years ago and oversees the strategic development for the molecular diagnostic segment. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Koene, you may now begin your presentation.
Thank you so much for the introduction. While COVID-19 is tightening its grip on the world, the WHO General Director has urged countries to test in order to control the infection rate and flatten the curve. Since January this year, many research organizations and in vitro diagnostic manufacturers have been scrambling to identify the coronavirus sequence and design diagnostic protocols and tests. Although the IVD industry is actively scaling up production to meet the sharp rise in demand, testing is still limited. No single organization is able to form a beachhead against COVID-19. Rather, it takes the collaboration within and across industries, as well as countries, to fight this pandemic. As a critical component supplier, LGC Biosearch Technologies plays a key role in supporting IVD manufacturers and clinical labs in this ongoing battle against coronavirus. This webinar will give you insight in our activities and capabilities. Biosearch Technologies is a comprehensive genomics portfolio from LGC, providing products and services for genomic analysis that support mission-critical application in molecular diagnostics. We support our customers from assay development to commercialization through our expertise in sample preparation, oligosynthesis, and components for PCR and NGS. Providing consistent quality and scale-up, we enable our customers seamless and fast time to market, delivering success at full scale from the start. As a true partner, we understand the needs of our customers along the complete product development pipeline and deliver solutions facilitating consistent quality at any scale. Through our cGMP manufacturing and ISO 13485 compliant facilities, we deliver vertically integrated raw material sourcing for critical components, ensuring global supply chain continuity. Our portfolio offers off-the-shelf, customized, and OEM products such as RUO and GMP oligos, including BHQ probes, high-performance PCR enzymes, isothermal enzymes, and master mixes, NGS reagents, nucleic acid components for DNA and RNA synthesis, sample preparation and purification chemistries, as well as instrumentation. Our recent Genome Web webinars with Co-Diagnostics provide a good example of how we collaborate with our customers, apply our capabilities and expertise to enable our customers to reach their goals. Thanks to the solid partnership we have built with Co-Diagnostics, We understand their processes, have short communication lines, and can support their needs accurately and in a timely manner. This is important for fast time to market, but even more crucial when responding to an outbreak, such as COVID-19. Having the capability to fast-track production scale-up while maintaining consistent quality and supply security is pivotal in these times. LGC Biosearch Technologies has a strong track record in responding to outbreaks, working closely with the CDC and molecular diagnostics manufacturers, providing components for molecular diagnostic tests, but also value panel reagents for swine flu, avian influenza A, and seasonal influenza. As we have learned from previous outbreaks and can see with the current coronavirus pandemic, the need for diagnostic tests increases rapidly. To meet demand, manufacturers and suppliers are required to quickly adapt and scale up production of these tests. With over 35 years experience in oligo manufacturing and a vertically integrated raw material sourcing, we are optimally equipped to scale up production and meet sharp increases in demand. Our customers, such as public health laboratories, value this as it enables them to meet their goals. Since the beginning of this year, we've mobilized resources and expertise to deliver rapid response to COVID-19 for customers in public health and private sector. We have been expediting oligos with BHQ technology for coronavirus testing, producing CDC-qualified lots of 2019 nCoV CDC probe and primer kits, and are currently working on making oligos and probes for other protocols available. We recently launched Seracare's AccuPlex coronavirus SARS-CoV-2 reference material and are actively testing new RT-PCR enzyme mixes such as EpiScript and the highly thermostable rapid fire. As we have been expediting oligo manufacturing for coronavirus testing, our portfolio of Fluorophores and quenchers, including our patented BHQ technology, form the backbone of many molecular diagnostic tests. With a broad range of dyes and highly efficient black hole quenching technology, we offer open platform solutions for targeted qPCR suitable for multiplexing with minimal crosstalk. Our range of BHQ probes, BHQ, BHQ Plus, and BHQ Nova, allows for flexibility in probe design based on the needs of your application. While our BHQ probes offer a cost-effective all-purpose solution, BHQ Plus delivers superior mismatch discrimination and BHQ Nova enhanced detection through optimal quenching. As the need for testing is increasing, we offer CDC-qualified lots of 2019 nCoV CDC probe and primer kits. Leaning on our 35+ years of oligo experience, vertically integrated raw material sourcing, and multiple manufacturing sites in the USA and EMEA, we can ensure supply quality and security while addressing flexibility in demand. Our operation process allows for timely and consistent upscaling capabilities, delivering 1 million tests per week with capacity to increase. With Seracare being part of the LGC group since end of 2018, we offer the AccuPlex technology, which enables full process control solutions. Full process controls mimic clinical samples far better than plasmids or naked DNA or RNA, as they must go through the extraction step to access nucleic acid for amplification, and they are highly stable. The recombinant alpha virus is genetically engineered to not replicate, and it is heat treated. It's therefore non-infectious and safe in use, as well as distribution. This technology delivers more consistent controls than inactivated virus and is customizable to sequences of interest. The AccuPlex technology was used to develop a recently launched AccuPlex SARS-CoV-2 product. The AccuPlex SARS-CoV-2 offers non-infectious and replication-deficient control, enabling safe handling. A full process control with real viral protein code. It is compatible with assays targeting CDC and WHO sequences as depicted in the current table. It also includes negative reference material. It has long-term stability at pragmatic temperatures, with no freeze-thawing required. And it is easy to adapt to unique assay designs or a potential new strain. The AccuPlex SARS-CoV-2 reference material kit comes with 5 vials positive and 5 vials negative of 1.5 milliliter each at 5,000 copies per ml. With a recommended storage temperature between 2 and 8 degrees Celsius, no freeze-thawing is required, providing ease of use for routine applications. Utilizing our probe and primer kit, as well as our AccuPlex reference material for SARS-CoV-2, we are actively testing our reverse transcriptase rapid-fire and EpiScript for one-step RT-qPCR detection of SARS-CoV-2. Preliminary results look very promising, and further details on this will follow soon. For many weeks now, the scientific community, industry, and healthcare are in overdrive to enable accurate diagnostic testing for SARS-CoV-2. As emphasized by the WHO General Director, to test, this is of crucial importance to flatten the curve of infection. However, whilst industry has been scaling up production, demand is still outstripping supply. LTC Biosearch Technologies continues to ramp up its activities in the fight against this pandemic. When looking for a component supplier, we recommend you remain selective and choose a supplier that truly works together with you. It is key to ensure there's a thorough understanding of your current needs and processes, as well as long-term ambitions, while maintaining supply quality and security. Identify how your supplier sources its raw materials and how it provides redundancy in manufacturing. Make sure the supplier is flexible and can meet change in demand through timely and consistent upscaling capabilities. Overall, it's key to build good partnership and short communication lines. I can recommend downloading the white paper, 6 Tips for Early-Stage MDX Companies Seeking Successful Commercialization, even if you're working in a clinical lab. As we all undertake our actions in the fight against coronavirus, we are stronger together. Our collaboration with Co-Diagnostics is a clear example of this. I invite you to reach out to us and explore how we can support you meeting your needs. Thank you all for attending. Stay safe, and the floor is now open for questions.
Thank you, Dr. Koene, for that informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Now let's get started. Our first question is, how can you support state public health labs who are working to develop their own laboratory-developed tests?
Yeah, that's a great question. We are providing a range of solutions that can be utilized by public health labs to develop their own LDTs. From custom oligos, RT-PCR enzymes, EpiScript, and rapid-fire to the CDC-qualified lots, as we just discussed in the presentation. as well as the AccuPlex coronavirus reference material. Considering the FDA's recently released policy guidance, high-complexity CLIA labs are permitted to begin using tests on patient samples as soon as they are validated internally, provided they have notified the FDA and subsequently file a EUA application within 15 days. This is also known as Policy A. Policy B enables U.S. states to authorize tests within their jurisdictions that will be run in high-complexity CLIA labs. The tests covered by Policy B can be used on patients as soon as they are validated and the FDA is notified. A subsequent EUA is not required in this case. Up to today, 4 states, Washington, Nevada, New York, and Maryland, have notified FDA of their intent to pursue this pathway. For detailed and up-to-date information, I recommend consulting the FDA's website.
Excellent. Our second question here is, how can your product support the ramp-up in COVID-19 testing?
Yeah, thank you for that question. We are a critical component supplier for many of the top MDX manufacturers and are really set up for high-scale production. Moreover, we are actively testing our individual components in order to provide a complete solution for molecular testing of coronavirus. Besides servicing IVD manufacturers, this will allow labs to develop and use lab-developed tests, which components for which supply is secured.
All right, and we'll be wrapping up with this final question. How do you ensure supply security with increasing demand?
Yeah, that's another great question. Not only are we set up for high-scale production, but through our vertically integrated raw material sourcing, we also have full control over our raw materials and have multiple production sites in both the USA as well as Europe, which really enables us to secure supply. Through our tight partnerships with customers, we're well informed on our processes and have short communication lines, and that really enables us to have efficient collaboration. And all that, taken into account, really helps us to ensure supply security.
Well, thank you again, Dr. Koene, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand through the end of 2020. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.